Welcome to American Medicine Today. I am Ethan Euchre here at the University of Central Florida College of Medicine campus, where they are doing some incredible breast cancer research. Let's check it out. So I'm here with Dr. Annette Khalid, the cancer assassin. I love that nickname, first off.
Dr. Khalid has been studying biomolecular science for over 30 years.
Let's kind of break down cancer. I mean, it sounds like a very basic thing to talk about.
Cancer is actually many diseases.
Okay.
It can affect different parts of your body, from your skin to your internal organs to your brain.
Very little is known about cancer cells. However, it is clear that the disease begins at the microscopic molecular level.
We do know, for example, that cancer begins small. It begins in
How small?
Undetectable.
Cellular level?
Cellular, molecular level. Molecular changes that happen inside of cells that are almost undetectable at this point. And we don't really understand what those changes all are, but we do know that it takes time.
Cancer develops due to the accumulation of molecular changes that occur over a period of time.
Cancer is not something that entered your body that's foreign, right? It's your own body.
It's not a disease that you catch from others. It's genetic or due to exposure to carcinogens that damage cells, causing them to behave in an abnormal way.
These things can damage cells and cause cells to behave in abnormal ways. They begin to grow. They begin to divide and create more copies of themselves, of the bad ones, right? Not good cells, but bad copies of cells. If you have liver cancer, your normal liver cells that are doing the function as they do for liver cells to keep you healthy become replaced with these abnormal cells.
So
and the good cells go away and the bad cells take over.
So eventually there's so many cancerous or bad cells that there's not enough good cells left to perform the function of that organ.
Exactly, so that organ now fails.
Metastasis is the process where cancer cells within a tumor leave the point of origin and move to a new location in the body. This leads to the patient now battling the disease in additional areas.
And now they're growing mini colonies of themselves in that new location. And as I was saying, they replace the good cells. And now instead of having just lung cancer, you have lung and liver cancer.
Some patients are able to successfully eradicate the cancer through treatment, only to have the devastating illness return later in life, and scientists don't understand the reason. This is why research is so important, as there is much we still don't comprehend.
So we don't understand all this. There's a lot of that's why we do research, because there's so much more that we need to learn about cancer.
And why do you think we're in 2018, 2019, we're in the 21st century, why do we still not know very much about cancer?
I think it's because we don't know a lot about ourselves still. We don't really understand. Our bodies are such complex machines. We don't understand at the molecular level how these machines work. And because cancer takes advantage of that and develops from these molecular changes that I was mentioning earlier, if we don't understand what the changes are still, we still don't understand what causes cancer.
While researching how general proteins work in our body, Dr. Khalid discovered a small piece of protein that behaved quite differently.
So we were studying this protein, as you mentioned, and just understanding how does it generally work? What's its general activity? Well, we came across this little piece of the protein that we were studying that by itself, it's only about 10% of the size of the total protein. This little piece had this own unique activity that was very different.
And what was this protein? Like, where is it found?
So this protein is called BAX, and it's found every cell of your body has BAX. And it's kind of a suicide protein.
BAX proteins travel to different parts of the cell after they die, and that movement is what led to her discovery.
Because we found that little piece of that protein actually had one activity when it was part of the protein, did one thing, but when you put it by itself, it had something completely, totally different. And what it does is it blocks another large protein that's a factory that helps protein folding.
Unlike other forms of cancer treatment, the peptide only disrupts cancer cells while causing little to no harm on normal cells.
Cancer cells need to fold to become like a key to fit into a lock of wherever they're traveling to.
Right.
And so your peptide interferes with that activity, so now it can't fold, and therefore it dies.
Cancer cells are super needy cells.
They sound like
They need materials, they need food, they need energy, but they especially need to have these materials properly folded, even more than our normal tissues do, than our healthy cells do.
The end of the BAX protein is the C terminus and is 20 amino acids long, leading Dr. Khalid to name the peptide CT20.
We're putting our peptide in a little capsule, but the capsule is nano size, which is
means
super, super, super tiny. I mean, I think You know, if you think of the head of a pin, you can put 1 million little nanoparticles on the head of a pin. They're super, super tiny particles. They're even smaller than cells. They're super, super tiny.
Dr. Flores first constructs the nanoparticles, starting with a polymer synthesis at the bio level. When the polymer is ready, she creates the nanoparticles to deliver the drug.
We start the process very early to the point that we start with the polymer synthesis. That part of the process, we don't do it in this type of lab because it's a bio level lab, but we work in collaboration with other professors in the university when we create this polymer.
Doctors use chemistry to create a polyester capsule that wraps around the peptide.
The fabric.
Some similar type of polyester. And it wraps around our peptide.
Okay.
And it forms these tiny little balls. And the chemistry allows, it's a very straightforward procedure, and the chemistry allows us to create a solution that has these little balls that captured our peptide and wrapped itself around it.
And that protects it from the things in the bloodstream that would destroy the peptide.
If you think about it, they're almost like, you know, they're like porcupines. These little particles, they have little things that come out of them that we can use like handles to attach things to. And so what we do is we attach things that tumors like to eat. And the tumors take them up, because that's the idea. And when they take them up, they basically ingest them. And then when that goes inside the tumor cell, that capsule breaks apart. So we know enough about what makes a tumor cell different from a normal cell that we can take advantage of that information and design our nanoparticles so they only are taken up by the tumor cells.
In cells, there's a factory known as the chaperonin, which folds linear proteins into three-dimensional shapes required for movement. This ability to travel is crucial for cancer cells to survive and spread.
So the chaperonin then folds all these proteins. So what our peptide does, it is basically blocks the works of the chaperonin.
Instead, they continue to produce linear proteins, which are toxic for cancer cells, causing them to shut down and die.
As a result, the cancer cells begin to accumulate all these linear proteins, because it didn't stop making protein, it's still making lots of proteins, but they're now all linear. And eventually that cancer cell will die from that.
Dr. Khalid's team has primarily focused on breast cancer, as this form is essentially a microcosm of all forms of cancer.
Because there's so many different types of breast cancer, it gives me the ability to study cancer as a whole.
Over the past year, they have started testing on other forms of the disease, beginning with small cell lung cancer, which has the highest amount of chaperonin and is therefore easier to test.
It's when the cancer begins to be more aggressive, becomes to be invasive, becomes metastatic, the chaperone levels go through the roof. And that's what we're able to target with our treatment.
Currently, Dr. Khalid and her team are researching the long-term effects of the delivery methods used on their cells.
But the nanoparticle is a challenge because the nanoparticle is another entity. You know, it's going to have, as I told you, we use it from a polyester polymer. Is that going to have an effect by itself? Could it cause side effects?
As in like the body rejecting it
exactly.
or
Could the body attack it? You know, it's a foreign particle. So,
right.
You know, so we need to understand how the body interacts with these nanoparticles.
In 2 years, UCF will be opening a new hospital, as well as expanding their cancer research center. Dr. Khalid hopes to have their CT20 ready for clinical trials by then, as they will have all the necessary resources right on campus.
There is a lot of excitement about nanotechnology that has died in the last few years. When at first the idea of nanotechnology was, hey, we can take my drug, my CT20 peptide, or any of the other drugs that are out there, and really reduce the dose because we can focus using nanoparticles that dose in tumors. Unfortunately, that idea is a wonderful idea, but it hasn't proven successful because of what we've discussed. The issues with these nanoparticles being taken up by different parts of the body, the nanoparticles, you know, being foreign particles that the body interacts with in a different way, so that we really haven't been able to show the promise of nanotechnology to deliver drugs like our CT20 peptide to tumors.
See, and they call you the cancer assassin. I don't know how you feel about that. I would be proud of it if it were me. How does that make you feel knowing that you could be, if not the person, part of a group that eventually solves one of mankind's biggest ills?
So we are the cancer assassins here. You know, if we can find ways to find cancers really early, our treatments are going to be even better. And even, you know, we're talking about cancer immunotherapy. Cancer immunotherapy works best early. The sooner that we can treat a cancer, the better off that patient will be.
The groundbreaking research being done by Dr. Khalid and her team of cancer assassins at UCF is continuing to bring the ultimate quest of eradicating one of mankind's most lethal killers into a reality.
Well, thank you for having us, Dr. Khalid.
It's my pleasure.
All right. Coming up next, a story of recovery.